Imagine, just for a second, that your laptop is severely glitching. Like the fan is screaming, the screen is flickering, and the operating system is essentially, attacking its own hard drive.
Yeah, that's a nightmare scenario.
Right. And you wouldn't try to fix that by downloading some massive unverified software update from like an unknown server on the internet, skipping the virus scan entirely and just hitting install.
No, you'd never do it. I mean, it's a
Right.
guaranteed way to completely crash whatever functionality you even have left.
Exactly. And yet, in the world of regenerative medicine, a very similar scenario is playing out inside the human body right now. Welcome to the deep dive. We have a highly specific, custom-tailored mission for you today.
We do, and it's a fascinating one.
It really is. We're exploring this rapidly emerging medical crossroads, specifically looking at the safety profile of vaginal exosome injections, and we are looking at this through a very particular lens.
Right, the autoimmune lens.
Yeah, exactly. What happens when these injections are given to patients who have an active autoimmune disease, like systemic lupus erythematosus or SLE?
It's just an incredibly high-stakes biological puzzle.
Yeah.
I mean, we're standing at this intersection where cutting-edge regenerative wellness basically collides head-on with a hyper-reactive, dysregulated immune system.
And the information out there, if you look for it, is giving people absolute whiplash. Because we pulled from a ton of sources for you today.
Yeah.
You've got direct consumer marketing from wellness clinics stretching from like Texas to Tokyo.
Yeah, they're everywhere now.
And they're promoting these treatments as safe, natural, cell-free miracles for tissue rejuvenation. But then, and this is the crazy part, you have stark public safety alerts from the FDA and the CDC detailing life-threatening complications.
Right, severe bacterial infections and sepsis. It's not a joke.
So to really understand the safety profile here, we can't just list off facts for you.
Yeah.
We need to actually understand the mechanics. So let's start at the foundation. Before we even bring lupus into the picture, let's establish what an exosome actually is. Sure.
Like, why are they being injected into intimate tissues in the first place?
Well, to understand exosomes, we have to scale way, way down to nanoscale biology. So exosomes are tiny, membrane-bound, extracellular vesicles.
Okay, when you say tiny, how small are we actually talking?
I mean, we are talking about 30 to 150 nanometers in diameter. They are microscopic, and they're released by almost all cells in the body. But for decades, scientists actually thought they were basically cellular garbage bags.
Wait, garbage bags?
Yeah. They thought it was just a way for cells to, eject metabolic waste.
But they're acting more like well, from the research, it looks like a localized postal service.
Exactly. They are microscopic messengers. They carry incredibly potent biological cargo packed inside this little lipid envelope.
What kind of cargo?
We're talking about specific proteins, lipids, growth factors, and highly targeted fragments of RNA, like microRNA.
Yeah, and these vesicles travel through the extracellular space, or the bloodstream, and they dock onto a recipient cell and transfer that cargo inside. And when they do that, they effectively rewrite the behavior of the receiving cell.
Which brings up a really important distinction, I think, especially when we talk about the marketing of regenerative medicine, because usually when people hear the word regeneration, they instantly think of stem cells.
Right, that's the big buzzword.
But exosomes are distinctly different. Stem cells are live cells. I mean, they can physically replicate and differentiate into new tissue types, which is powerful. But installing live cells carries the risk of those cells replicating out of control.
Yeah, forming tumors or, triggering immune rejection.
Right, but exosomes are entirely cell-free.
And that cell-free characteristic is the absolute bedrock of the marketing you mentioned earlier. Wellness clinics argue that because there are no live cells to replicate, the safety profile is just inherently better.
Like it's idiot-proof.
Essentially, yeah. They say you're getting the regenerative signaling of a stem cell without the unpredictable baggage of a live cell.
So going back to our computer metaphor from the start of the deep dive, if a stem cell is like physically installing a brand new piece of hardware into your computer with all the, compatibility risks that brings an exosome, is just the software update.
That's a great way to visualize it. It's literally a packet of code that tells your existing hardware to run differently.
Okay, so clinics are utilizing these biological software updates for what they broadly call vaginal rejuvenation.
Right. Because as women age or experience profound hormonal shifts like menopause, the tissue in the vaginal wall can become atrophic.
Meaning it breaks down.
Yeah, the tissue physically thins out, it loses its elasticity, and crucial vascularity just declines. And this leads to severe discomfort, dryness, and a real decline in sexual function.
So the mechanics of the treatment involve injecting these exosomes as submucosal microdroplets directly into the vaginal wall.
Yes, right into that thinning tissue.
And the goal here is extracellular matrix repair. But what is the exosome cargo actually doing on a cellular level to fix that?
Well, the extracellular matrix is the scaffolding of the tissue. In atrophic tissue, that scaffolding is actively breaking down faster than your body can rebuild it. The exosome cargo is designed to reverse that ratio.
How so?
First, the microRNA and proteins inside the exosome downregulate specific enzymes called MMP1 and MMP3.
These are the matrix metalloproteinases, right?
Right.
They basically act like molecular scissors that chop up structural collagen. So step one of the software update is basically turn off the molecular scissors.
Exactly, stop the breakdown. Then step 2 is to start the builders. The exosome cargo induces local fibroblasts, which are the cells whose whole job is making connective tissue to generate brand new collagen and elastin.
Okay, that makes sense.
But perhaps more importantly, the exosomes stimulate neoangiogenesis, which is the creation of new blood vessels. And they do this primarily by activating a cellular signaling route called the AKT-mTOR pathway.
Oh, wait, I've seen mTOR mentioned a lot in longevity research. How does activating it here actually help with atrophic tissue?
So the AKT-mTOR pathway is essentially a master switch for nutrient sensing and cell growth. When the exosome cargo flips that switch to the on position inside the endothelial cells, those cells start dividing and forming new capillaries.
Oh, wow. Okay.
And more capillaries mean more blood flow, more oxygen, and ultimately thicker, healthier mucosal tissue.
Stop the tissue breakdown, build new collagen, and restore blood flow. Mechanistically, I mean, it sounds brilliant. You can easily see why a patient struggling with severe atrophy would be totally drawn to a clinic offering this.
It sounds like magic on paper.
It does. But we have to introduce the critical variable for our deep dive today.
Okay.
We know how this biological code is supposed to execute in a healthy person. What happens when you introduce this intense cellular signaling into a body where the immune system is already completely misfiring?
And that brings us to systemic lupus erythematosus. Lupus is a highly complex autoimmune disease characterized by a fundamental loss of immune tolerance.
Meaning the body gets confused.
Right. The patient's immune system forgets how to distinguish between self and a foreign invader. So it produces autoantibodies that relentlessly attack the body's own healthy tissues and organs.
And the relationship between lupus and exosomes is incredibly complex based on the research. It's almost a complete duality, because on one hand, we're seeing that the patient's own exosomes are actually a primary driver of the disease.
Yes. During an active SLE flare-up, the circulating endogenous exosomes, so the ones the patient's own body is making, they become fundamentally toxic.
Toxic how?
They are loaded with specific inflammatory microRNAs, particularly miR-21 and miR-146A. And when these toxic exosomes interact with the patient's immune cells, those specific microRNAs happen to chemically mimic viral RNA.
Wait, let me make sure I'm getting this right for the listener. The body is packaging up its own microRNA, sending it out, and the receiving immune cells look at it and literally think the body is under a massive viral attack.
Yes, exactly. The immune cells have these internal sensors called toll-like receptors, or TLRs. You can think of them as the immune system's internal smoke detectors, specifically calibrated to detect the genetic signatures of viruses.
Okay.
And when those smoke detectors get tripped by the patient's own exosomes, it triggers a massive cytokine storm.
So the alarms go off, and the immune cells start flooding the surrounding tissue with inflammatory proteins, things like interferon alpha, TNF-alpha, and IL-6.
Right. It creates this devastating cascading loop of severe systemic inflammation. The tissue damage you see in lupus isn't just a byproduct, it's the direct result of these endogenous exosomes constantly screaming virus to an already hypervigilant immune system.
Okay, but then there's the other side of the coin, because we see immunological research where scientists are desperately trying to use engineered exosomes to cure lupus.
They are, yeah.
They're taking exosomes derived from mesenchymal stem cells, MSCs, and using them to actually calm the immune system down.
The therapeutic potential of MSC-derived exosomes is just staggering. In preclinical models, these engineered exosomes can modulate T cells and actually switch the behavior of macrophages.
Macrophages being the white blood cells, right?
Right, they're a type of white blood cell that can exist in different states. The engineered exosomes can actually force them to shift from an aggressive, inflammatory state, which is known as M1, into a healing reparative state known as M2.
There's also that cutting-edge research using NK92 cells. The researchers engineered natural killer cell exosomes, equipped them with a CD19 targeting mechanism, and used them in a mouse model of SLE.
Oh, that study is incredible.
It is. They essentially created a microscopic guided missile that specifically targeted and depleted the malfunctioning B cells. The survival rate of the treated mice jumped to about 80%, compared to just 25% in the untreated group.
It's an incredible demonstration of how specific and powerful exosomal cargo can be when engineered precisely in a laboratory setting.
Right, in a lab setting. But hold on, I'm looking at this whole picture and something just isn't tracking for me.
What do you mean?
If a lupus patient's own exosomes are already acting like malicious code, constantly tripping the viral smoke detectors and spreading a cytokine storm, why on earth would anyone think injecting a massive, unverified dose of foreign exosomes into highly vascular, intimate tissue is a good idea?
Yeah,
Isn't that just throwing biological gasoline on a fire?
that is the exact crux of the issue. You are identifying the massive, dangerous gap between tightly controlled, highly engineered preclinical lab research and the reality of the broad-spectrum exosome products currently being sold to consumers in the open market.
Which brings us to the reality check. The marketing promises versus the actual Wild West of regulation. When you look at the websites for these wellness clinics, the messaging is incredibly reassuring.
Very polished.
Yeah, they promise minimal downtime. They list the side effects as nothing more than temporary mild redness, a bit of puffiness, or maybe minor bruising that fades in a few days. They market it as a lunchtime procedure.
But look at the regulatory side. The FDA, the CDC, and organizations like the International Society for Extracellular Vesicles paint a completely different picture.
A very dark picture.
Exactly. The FDA has unequivocally stated that there are currently no FDA-approved exosome products for treating diseases or conditions in humans. None.
Wait, none? If there are no FDA-approved products, how are these vials of exosomes even being manufactured and ending up in high-end strip mall clinics?
This is a massive regulatory blind spot. Because they aren't approved drugs, there are zero standardized enforced protocols for how these exosomes are extracted from donor tissue, how they are purified, or how they are preserved.
Okay, what does that look like on a cellular level? Like, how do different extraction methods actually change the product you're buying?
Well, let's compare two common methods. In a high-end, rigorous lab setting, they might use something called tangential flow filtration, or TFF. This uses porous membranes and gentle fluid dynamics to separate the exosomes from cellular debris without damaging their incredibly delicate lipid envelopes.
Okay, tangential flow filtration sounds a bit like using a gentle French press to separate coffee grounds from the liquid.
That's a really good visual, actually. Now contrast that with ultracentrifugation, which is what many less sophisticated facilities use because it's significantly cheaper.
Okay, what is that?
They spin the source material at upwards of 100,000 times the force of gravity.
Oh, wow. So instead of a gentle French press, they're basically throwing the whole thing in a blender on high speed.
Exactly. And the result of that sheer physical force is that a massive percentage of those delicate exosome vesicles are ruptured. And when they rupture, they spill their microRNA and protein cargo prematurely into the solution.
So they're broken.
Yeah, you aren't getting intact messengers. You're getting a soup of free-floating genetic material and broken lipid membranes.
And without standard USP or ISO manufacturing oversight, I mean, the risk of flat-out bacterial contamination skyrockets. Like the CDC investigated a terrifying cluster of cases in Nebraska where patients received unapproved exosome interventions for joint pain.
Yeah, I remember reading that report.
They ended up hospitalized with severe bacterial infections, and some developed life-threatening sepsis. And there have been similar reports out of Japan involving serious adverse events following unregulated exosome administration.
The burden of verifying the safety and sterility of these products is falling entirely on the consumer. And for a completely healthy individual, the risk of receiving a ruptured or poorly processed exosome product is already significant.
Right.
But when you introduce the variable of an active autoimmune disease like SLE, the safety profile shifts from risky to highly precarious.
So let's synthesize all of this mechanistic data specifically for you listening. When we combine the local physical realities of an intimate vaginal injection with the systemic hyperreactive reality of lupus, what is the ultimate safety profile? Let's start with just the physical tissue.
Well, physically injecting anything into atrophic vaginal tissue requires extreme precision. The tissue is thinned out, it lacks estrogen support, and the capillary beds are vascularly fragile.
It's dealt.
Very. When a practitioner injects submucosal microdroplets of fluid into that compromised environment, there's a very high risk of localized hematoma. You are essentially creating trauma, which leads to severe bruising and blood pooling under the mucosal lining.
But a healthy person might just get a localized bruise and recover in a few days. A patient with active SLE isn't just dealing with fragile tissue, they are dealing with the drugs required to survive lupus. They are heavily reliant on high-dose immunosuppressants or corticosteroids.
Right, and that radically changes the risk equation. Those medications are deliberately suppressing the immune system's ability to fight off invaders, to stop it from attacking the patient's own organs.
Yeah. So if a clinic is using an exosome product that lacks rigorous manufacturing standards, or if someone simply left a box sitting on a loading dock and broke the cold chain storage.
The introduction of a contaminated vial is no longer just a localized infection risk. For an immunosuppressed SLE patient, a minor bacterial contamination that healthy immune system might just wall off and neutralize. It can rapidly escalate into systemic, life-threatening sepsis.
Because the body simply doesn't have the white blood cell response required to fight it back.
Exactly.
Okay, let's play devil's advocate for a second. Let's assume the clinic is operating flawlessly. The exosome vial is 100% sterile, CGMP certified, and they use the gentle French press method so all the vesicles are perfectly intact. Is it safe for a lupus patient then?
Even under those absolutely perfect conditions, immunological theory suggests extreme caution because exosomes are defined by their surface markers.
Okay, explain that.
The outside of the lipid envelope is studded with specific proteins called tetraspanins. These are markers like CD63, CD81, and CD9.
So even if the cargo inside is designed to be healing, the outside of the envelope still has a chemical signature.
Yes. You are taking millions, potentially billions, of foreign exogenous vesicles derived from another human being's cells and dropping them directly into an immune system that is already severely dysregulated.
Going back to those toll-like receptors we talked about, the viral smoke detectors. In a lupus patient, those detectors are already highly sensitive. They're already misinterpreting the body's own signals.
Exactly. Introducing that dense concentration of foreign biological markers could theoretically be the exact trigger that sets off an unpredictable, severe autoimmune flare-up.
Oh, wow.
Because, to a hypervigilant immune system, those millions of foreign tetraspanins might as well be a neon sign saying massive pathogen invasion. The immune system won't perceive those exosomes as a healing software update. It will perceive them as a massive threat, triggering the exact cytokine storm the patient is trying to avoid.
The potential reward is undeniably alluring, though. I mean, you're talking about rejuvenating painful, atrophic, intimate tissue, restoring vascularity, and maybe getting a piece of normal life back. But the mechanics we've walked through show a pretty terrifying reality.
It's very risky.
The ammunition, the exosome product, is entirely unregulated, prone to rupture and contamination. And the target, the SLE immune system, is incredibly volatile. The danger of a biological misfire is just exceptionally high.
It really is. While there are legitimate, high-quality exosome products in rigorous research environments showing low immunogenicity, the current lack of industry standardization across consumer wellness clinics means an SLE patient is taking a significant, unquantifiable risk.
You just don't know what you're getting.
Right. The variables are simply too wild to predict how an individual's dysregulated immune system will react.
So to sum up what we've unpacked for you today, exosomes are undoubtedly powerful. They are natural biological messengers with incredible proven potential for both treating autoimmune diseases in a lab setting and regenerating intimate tissues.
The underlying science of the AKTMT pathway and extracellular matrix repair is very real.
Right. But right now, the total lack of clinical regulation, combined with the extreme sensitivity of an active SLE immune system and the physical fragility of atrophic vaginal tissue, means the safety profile of these intimate injections is highly precarious.
Highly.
It is fraught with localized physical risks, profound contamination dangers due to immunosuppression, and the theoretical threat of triggering a systemic immune flare.
It is a classic case of the commercial wellness market moving years ahead of the actual clinical safety data. But I don't want to leave this entirely in the dark because if we look to the future, the science of exosomes offers a really provocative path forward for autoimmune diseases like SLE.
Because earlier we talked about how a major driver of lupus is the patient's own toxic exosomes, the ones carrying that miR-21 cargo spreading inflammation.
Right. So perhaps the ultimate future of exosome therapy for SLE isn't about going to a clinic and injecting new foreign exosomes into the body to try and force the tissue to heal.
What if instead the treatment is about getting rid of the bad ones?
Exactly. Researchers are theorizing about developing a way to essentially scrub or filter the patient's existing exosome network.
Oh, that's fascinating.
Think of it like a highly specific nanoscale dialysis. But instead of filtering the blood for metabolic toxins, you are circulating the plasma to specifically filter out the endogenous exosomes carrying that inflammatory viral mimicking cargo.
So instead of risking a total system crash by installing an unverified software update from a sketchy clinic,
Right.
the future of autoimmune medicine might just be running a highly targeted antivirus program to clear the biological malware that's already there.
And removing that constant inflammatory signal would allow the body's own healthy baseline systems to finally reboot and heal the atrophic tissue naturally, without the risk of foreign biological materials.
Now, that is a software update I think we'd all be willing to install. It's a fascinating paradigm shift
All right.
and something to mull over as we watch this incredible field of nanoscale medicine evolve. Until next time, keep questioning the code.